Again, good morning, good afternoon, good evening. Welcome to this webinar, and thank you very much for attending. So, as always, I will make a very short update on what the Global Virus Network is about. Some of you may not be aware of it, although I know that most of you are aware. And then a short introduction to our two speakers, and then we will have the lecture. So the Global Virus Network is a science-driven, independent network now of 70 research centers and 11 affiliates worldwide. Our missions really includes research, education and training, and advocacy communication. We have task force on different viruses, of course, very much focused on COVID-19, but actually working on all human viruses. And we have a very strong program on education and training that you can find on the website with the GVN Academy that we have created. And we have a strong focus also on advocacy and communication. Regarding COVID-19, I just want to provide the information. We will send the announcement very soon that the GVN will hold a very important international conference on long COVID, which, as all of us know, is a very important challenge, on July 20 and 21st, to be held in Baltimore, in person and as a hybrid by video conference, and we will provide all the necessary information. So now it's really my pleasure to introduce our two speakers. We will have two speakers today. Céline Cougoule, who is a senior research scientist. She's a CNRS from France, CNRS Research Associate at the Institute of Pharmacology and Structural Biology in Toulouse, in France, which is a very high-level research institute. She's working in a team focused on immune detection and elimination of pathogens, investigating how host and pathogen mechanisms can control the autonomous immune response of stromal cells. And she has developed a lot of preclinical models she will speak today, I guess, about. And our second speaker is Nicolas Hoertel. Nicolas is an MD, PhD. He is an Associate Professor of Psychiatry in Paris at Paris Cité University and Corentin-Celton Hospital. He holds a PhD in Neuroscience at Paris Descartes University. He has also been a research fellow at the New York State Psychiatric Institute, and he has a number of degrees, including a Master of Public Health. And his research interest has been focused on epidemiology and pharmaco-epidemiology in the field of mental health, with an emphasis on old age psychiatry, psychiatric and medical comorbidity, psychotropic medications, and suicide prevention. Now, today, both of them are going to present some, say, provocative findings on the association between the use of certain antidepressants and evolution and prognosis of COVID-19. So thank you very much, both of you, for giving this lecture. And I guess that we will start with Nicolas Hoertel for his presentation. Thank you.
Thank you very much. I will share my screen.
Yeah, we can see it.
Perfect, thank you. First of all, along with Dr. Céline Cougoule, I would like to deeply thank you, Professor Christian Brechot, for this invitation, and also to thank the whole Global Virus Network staff. It's really an honor and a great pleasure to present to you recent findings on especially two antidepressants, two SSRI, fluoxetine and fluvoxamine, for COVID-19. So here are our conflict of interest. So I think for all people connected, I think you all heard about specific antidepressant called fluvoxamine, which made the headlines of great literature journals, Science, Scientific American, and Nature, following clinical trial, randomized clinical trial that published positive results using this molecule against COVID-19. It had already made headlines of the general press media, I think worldwide, in France as well as in the United States. So the purpose of this lecture is to go back in time and to try to reconstitute both the scientific story, because it implies many researchers worldwide, and to try to describe also the methodology employed to try to see whether this medication could be efficient as a repurposed medication against COVID. So shortly, this story will discuss the first observation that our team did about this association, the potential association between the use of certain antidepressants and the better prognosis in hospitalized patients with COVID-19. Then these results gave birth to an hypothesis that certain antidepressants could have both antiviral and anti-inflammatory properties through a specific mechanism which implies the sphingomyelin acid and ceramide system that we will detail. This hypothesis has been confirmed in an animal model that Dr. Céline Cougoule will present the details. And we are at the stage where we may have found a potential biomarker predictive of COVID-19 severity. And with ongoing clinical trials testing both fluoxetine and fluvoxamine in Africa through the anti-COVID clinical trial conducted by the DNDi, but also other clinical trials in Brazil conducted by Edward Mills with the Together trials and other trials worldwide. First, I'm working in a specific department headed by Professor Frédéric Limosin in France. More precisely, it's near Paris, a city called Issy-les-Moulineaux. And in our department, in our medical center, which has 80 full-time beds and specialized in all-day psychiatry, and particularly in mood and anxiety disorders for older people, since February 2020, we only had 3 cases of severe COVID-19. So we're at the beginning of the pandemic. We were highly surprised because colleagues from other departments, and especially from geriatric departments, were reported that once one patient were infected, often the full floor of patients had many cases in the few days after the detection of the first case. First, we wanted to understand whether this empirical observation could be generalized and whether other colleagues had the same observation that our patients with psychiatric disorders, at least some of them, and especially older adults, could be at lower risk to have COVID-19 or to have severe form of COVID-19. So questioning this link between psychiatric disorders and COVID-19 outcomes. So first of all, we performed an observational study using electronic health records from AP-HP, Paris University hospitals. And we examined a sample of near than 50,000 patients hospitalized for laboratory confirmed COVID-19. And we examined the prevalence of people with any primary psychiatric disorder, including also addictive disorder and neurocognitive disorder. And we were surprised to see that this prevalence was only, if I can say, 3.4%. Then we examined the association among these patients with the primary psychiatric disorders, the prognosis in terms of mortality. And we found that the crude association was significantly associated. So there were few people, if I can say, having a psychiatric disorder, but these people seemed to have a lower prognosis. But when adjusting for age and sex, this association was strongly reduced. And when taking into account medical comorbidity, I mean, other medical conditions, we were surprised to see that we reversed this association, meaning that these people with psychiatric disorders hospitalized in our centers had a very high number of comorbid medical conditions. In mean, it was 12 medical conditions as compared to 4 medical conditions in COVID-19 in patients without any psychiatric disorder. This observation was also of prevalence of psychiatric disorders among people diagnosed with COVID-19 was supported by a systematic review of 33 epidemiologic studies published in the Lancet Psychiatry earlier in the pandemic that found that only 2.98% of people with COVID-19 had psychiatric disorders. In contrast, just to have an idea, even if it's always difficult to do a direct comparison, in contrast, the World Health Organization has estimated that 15% of older adults suffer from at least one mental disorder. And finally, a study conducted in Germany in a sample of more than 8,000 hospitalized COVID-19 patients found that depression was associated with reduced 6-month mortality with an effect size that was substantial with an odds ratio of 0.46 when adjusting for sex, age, obesity, as well as other medical conditions. So could antidepressants a priori, theoretically, be useful in COVID-19? In fact, antidepressants are repurposed medication. The first antidepressant was iproniazid, an antibiotic that was designed to fight tuberculosis. In fact, it has been observed that even if it was not very efficient in people with tuberculosis as a sole treatment in sanatorium, clinicians observed that these people had a better mood. And from this observation came the idea to propose to depressed people this medication, and it was the first antidepressant. What is interesting is we have already a substantial literature showing that antidepressants do have anti-inflammatory properties in individuals with major depressive disorder. Here is the meta-analysis of this study showing that antidepressants used are associated in a before-after design with significant reduction in several inflammatory markers, interleukin-6, TNF-alpha, interleukin-10, and CCL2. And what is interesting and striking for us at the beginning of the epidemic is that these inflammatory markers were reported as associated with severe COVID-19. Second, we had 2 animal models, specifically 2 LPS-induced sepsis mouse models, one with fluoxetine, another more recent with fluvoxamine, that shows that fluoxetine and fluvoxamine are associated with reduced mortality in this sepsis mouse models and reduced levels of certain inflammatory markers, including TNF-alpha for fluoxetine and interleukin-6 and interleukin-1 beta for fluvoxamine. The exact mechanisms underlying the anti-inflammatory properties of antidepressants are not well known, but we have several hypothesized mechanisms. One of them is the sigma-1 receptor agonist effect, which has been consolidated in this mouse model, showing that it could be involved in these anti-inflammatory properties. I'm recalling here below the order of the effects of different antidepressants on sigma-1 receptors, because all antidepressants are not acting the same, with fluvoxamine the best one, the second one fluoxetine, and then escitalopram, citalopram. This ranking is based on studies in brain of rats. Other potential mechanisms are non-sigma-1 receptor pathways. It could include NF-kappa-B, inflammasome, TLR4, PPAR gamma. Other mechanisms could also include immune modulation, reduced platelet aggregation through reduced serotonin uptake, antioxidant properties, decreased mast cell degranulation, decreased melatonin degradation. And finally, chiasma effect, and we will focus on that later, acting in endothelial cells and the immune system that could also potentially contribute to these anti-inflammatory properties. So at this stage, now, the hypothesized mechanisms of certain antidepressants that might be useful in COVID-19, including anti-inflammatory effects, as well as antiviral effects. This hypothesis was that it could also be driven by the chiasma effect through the inhibition of the SM activity in upper airways and lung cells. And another mechanism is the interference with lysosomal trafficking. We'll focus on the chiasma effect very soon. So first, we wanted to, after this observation, we wanted to test the effect of these antidepressants with observational data as a first step. And we conducted the first observational study showing that antidepressant use was associated in our electronic health records with reduced risk of death or intubation in patients hospitalized for COVID-19. The study included more than 7,000 adults, of which 345 patients were receiving an antidepressant at baseline, approximated by 48-hour of hospital admission. The mean fluoxetine equivalent dose was 20 milligrams per day. And we adjusted our analysis for patient characteristics, clinical and biological severity markers at baseline, and other medications. So this association was substantial because we found that any antidepressant was, if I can say shortly, was dividing by 2 the risk of death or intubation in these patients. And we were surprised to see that it concerns both SSRI, so serotonin selective reuptake inhibitors, and non-SSRI antidepressants. And more specifically, 5 molecules were individually significantly associated with this outcome. It was fluoxetine, escitalopram, paroxetine, venlafaxine, and mirtazapine. Of these 5 molecules, 3 had very, very low or inexistent sigma-1 receptor effects. We wanted to confirm this observation with a stronger design, observational design, and wanted to examine whether antidepressant use could have antiviral properties in the way that people taking antidepressant could be less likely than expected to be hospitalized with COVID-19. And the second aim was to retest whether antidepressant use was associated with reduced 28-day mortality in patients with COVID-19, using a different sample as we used for the prior study. We included more than 380,000 inpatients tested for COVID-19. It was very easy because, as you know, and as in many hospitals, patients during the pandemic waves were systematically tested at admission. So we had some direct tests to be able to see whether this patient tested positive or negative to SARS-CoV-2 infection. Out of this population, more than 40,000 patients had a confirmed, laboratory-confirmed COVID-19. For the first aim, we did a matched analytic sample and took for each patient with laboratory confirmed COVID-19, 1 patient sharing the same characteristic in terms of age, sex, hospitalization period, and number of medical conditions. And we found that that antidepressant use was less prevalent in patients with COVID-19 than those without COVID-19, knowing that these patients were sharing the same characteristic as listed before. And the gap was huge because the odds ratio was 0.38 and highly significant. The second aim, we wanted to examine whether people with laboratory confirmed COVID-19 had a lower 28-day mortality when having at baseline an ongoing prescription of antidepressant versus not. And it was also significant with an odds ratio of 0.55, and interestingly, with a significant dose-effect relationship. As you can see, for doses lower than 20 milligrams, we didn't have a significant difference, a tendency, but not a significant difference as compared with no antidepressants, but it was significant when for doses of 20 milligrams or more, and even more significant and more pronounced for at least 40 milligrams per day of fluoxetine equivalents. Then we examined other studies, and there have been numerous studies examining this association between antidepressant use and infection and mortality. And so I tried to summarize these studies and the effect sizes, but at the same time, we know that observational studies are the subject of multiple biases. And I wanted to examine some of them for all published studies. First, if COVID-19 was confirmed, we know that some studies, clinical appreciation was sufficient, or and other studies, inclusion criteria included the fact to have a laboratory confirmed diagnosis. Whether the observational study was taking into account the medical condition that we observed as very, very frequent and more frequent in people taking antidepressant and hospitalized for COVID-19. Whether they measured dose, I think it's something very important because in electronic health records, we know that the software can detect fluoxetine as a keyword, but it could be a past prescription and not a current prescription. Having the possibility to having the dose is reinforced the likelihood that people are actually taking antidepressant. And the last bias is the immortal bias to see whether it was they were taking into account the ongoing or baseline antidepressant use and not during the hospitalization, which could, of course, create biases. And globally, what we found, there are two studies that examine the risk of infection, and both studies indicated significant reduction of COVID-19 detection, so supporting possibly the antiviral effect, meaning that people can be infected with COVID-19, but they may not reach a sufficient load of virus in the nose to be detectable by PCR. It could be this hypothesis. The second one is that many studies found decreased also all-cause mortality, except several studies here, including one that showed potentially increased mortality. But as you can see, these studies had several important biases, which limits the interpretation of these findings. Other studies showing protective association had, at least for the specified limitations, had lower risk of biases for these points. Of course, we know that observational studies are not a sufficient level of proof, and the classical pyramid of proofs places observational cohort studies in the middle of proofs. However, what is interesting is this paper in the New England Journal of Medicine that did a great job that compared observational studies and RCT's results for the same medication, for the same indication. And what they concluded, here I took the example of this paper of the odds ratio for infection after laparoscopic as compared with open appendicectomy, is that for good observational studies, if I can say, I mean for well-designed observational study, the global conclusion and effect size is generally near to that of what is obtained with randomized control trial combined. And the last thing is what Henri Poincaré, along with Marie Curie here, recalls us in methodology that, of course, replicability is key, that it's not just one perfect trial that could definitively give the solution or give the truth that the replicability of well-designed studies by independent teams that increase our confidence in scientific evidence. So now let's talk about the Fiasma hypothesis. Very shortly after the publication in the preprint of our results, observational results, we've been contacted by two professors, Professor Gelbins and Professor Hoertel from Germany. They said that our results were compatible with the fact that the specific antidepressants that was associated with reduced intubation or death in our study were those who had the highest effect on acid sphingomyelinase. So what is acid sphingomyelinase? It's a lysosomal enzyme that, following a stimulus, translocates into the membrane of the cell. And this enzyme is responsible of the degradation of sphingomyelin, a sphingolipid, into phosphorycholine and ceramides. And ceramides, the accumulation of ceramide results in fusion of rafts and receptor clustering, able to concentrate at a specific point of the membrane, receptors responsible to increased signal transduction. And Fiasma, how do they act? Fiasma, like several antidepressants, are entering the lysosome and are detaching the ASM into the middle of the light of the lysosome, and the ASM is destroyed and then inactive when coming in the membrane to be able to create these rafts and receptor clustering, so blocking the signal transduction. They tested this hypothesis right away and examined whether with pseudo-particule of protein spike was able to trigger the activation of this enzyme. And it was positive and specific to these pseudo-particles. Then they tested in vitro, in Vero-E6 cells, classical, the antiviral effect, and found that it has, this antidepressant have strong antiviral effect. But what they did also, they examined whether they were able to restore this infection in vitro by adding a ceramide and the C16 ceramide, and they observed the restoration of the infection. And finally, they go further and did an experiment in healthy volunteers. So the experiment was to take nasal cells and to put them in contact with the virus, pseudoparticles, and they observed infection, direct infection. And then they gave to these healthy volunteers one single pill of an antidepressant called amitriptyline, a very strong Fiasma. And 1.5 hours after taking this pill, they took these nasal cells and examined whether these nasal cells were infectable. And they observed that there were no infection. And 24 hours after taking this pill, they examined the nasal cells and observed restarting of this infection. Here we can see a little bit more, supporting that maybe we should just take another pill. So to summarize this finding, the hypothesis is that the virus, when connecting to its ACE2 receptor, induced the stimulation and the activation of this enzyme, which creates the ceramides, which may constitute the entry door into the cell. By blocking the acid sphingomyelinase by a Fiasma molecule, they close potentially this entry door. After these results, we had the idea to test this hypothesis with our observational data. So what we did is we compared antidepressants with high versus lower Fiasma activity in terms of intubation or death in COVID-19 in patients with psychiatric disorders to decrease the indication bias. And it worked, because we saw that this risk was significantly reduced by almost half in all our analyses, and with different approaches and full sample and matched analytic sample. We also observed that the prevalence of antidepressant, which was found to be lower in COVID-19 patients taking COVID-19 in patients as lower than expected, given the age and comorbidities, and that this lower prevalence were more pronounced for antidepressants with higher chiasma activity. By the way, we found, contrary, we found no significant difference across sigma-1 receptor agonism for this prevalence of antidepressants. So if this theory is true, we may have found a biomarker, and I mean Professor Galbins and Conduber may have found a biomarker predictive of COVID-19 severity. And in fact, it has been already tested. The question was whether the levels of ceramides in the blood could be predictive of the severity, the clinical severity of COVID-19. And what they found is the higher is the levels of ceramide in the blood, the worse is the prognosis for these patients. Also, another experiment was found to compare different degrees of severity of the disease as compared with convalescent patients. And we saw a kind of dose effect, if I can say, across the severity levels in these patients. 4 studies have concordant findings for that. And also 2 studies found a correlation with ceramide levels and inflammation levels in the blood of COVID-19 patients. So what medication do have this chiasma activity? In fact, it includes both several antidepressants, not all antidepressants, specific antidepressants, but also other non-antidepressant molecules. But we already know that, in fact, not all chiasma medication could work. Why? First, because expected in vivo chiasma effect may depend on the in vitro activity, in vitro chiasma activity of these molecules. We know that it may vary very strongly across molecules. Second, it depends on the dose prescribed, of course, but also of the tissue distribution, and especially the lung and brain distribution, which may differ, we know, to blood half-life. And I wanted to show you, so it's the list of medication that showed in vitro chiasma effect, that we already have data for several molecules, in vitro data showing this antiviral effect. So regularly, we're seeing in the literature other teams saying, okay, we found another molecule with antiviral properties. And often it was, okay, it was a chiasma. And we had that for this molecule with the green circle. But when we also look at the studies that reported positive observational data. And we see that we have a lower number of molecules. Most of them were antidepressant, but also antihistaminic medication like hydroxyzine and amlodipine, a calcium channel blocker. So we also tested with our observational study and found that chiasma medication were also associated with lower intubation or death in more than close to 3,000 adults in patients hospitalized for severe COVID-19. And these results were mainly explained by chiasma antidepressants, hydroxyzine and amlodipine in our analysis, since these treatments were representing most chiasma treatments. So if we are looking at what are the best, if I can say the best in vitro chiasmas, in fact, the 2 best ones are amitriptyline and fluoxetine. And especially among antidepressants and among SSRI. And what has been tested is whether the antiviral effect was correlated with the intensity of inhibition of the acid sphingomyelinases. And as you can see, there is a strong correlation. We were not interested, in fact, even if it's a great antidepressant, a great chiasma, I want to say that we were not interested that much by amitriptyline because it's a tricyclic antidepressant. And tricyclic antidepressant, an old antidepressant, is not well tolerated in older adults. So right away, we know that this molecule is not a good candidate for the next step. But SSRI are pretty well tolerated among the older adults. And in fact, fluoxetine is the best tolerated and the best plasma, I mean, in vitro speaking, among all SSRIs. So we are highly interested by this molecule. And as you can see, fluvoxamine is also a plasma and ranked the third one among SSRI. So what molecule to test in priority? Those with known good tolerability, if possible, easily available and cheap to allow its use in resource-poor countries, the strongest in vitro plasma effect, with high lung and brain concentration to shortly reach at a standard non-toxic dose a plasma effect in these target organs. Based on this characteristic, we thought that fluoxetine and fluvoxamine are very good candidates. But specifically for fluoxetine, what's the optimal dose to test in a clinical trial? We know. So how to reason? For fluoxetine, we have specific data. First, we have in vitro data on Calu-3 cells on antiviral effect with the EC90 at about 4 micromolar. And we also have experimental postmortem data showing that lung concentration of fluoxetine is 60 times higher than blood concentration, allowing to do a population PK modeling to try to predict the proportion of patients taking this molecule that would reach a lung antiviral effect, a lung plasma effect. And they showed that at 40 milligrams per day, 55% of patients of the population are likely to reach at 24 hours this effect, and 87% at 72 hours. And this dose is compatible with the recommended dose of fluoxetine for depression in both adults and in the elderly, that is between 20 and 40 milligrams. This idea was reinforced by our observational data showing a dose effect of fluoxetine specifically on 28-day mortality. And as you can see, the mortality was lower for people taking at least 40 milligrams. In fact, it includes 40 milligrams, compared to people taking less than 40 milligrams. And that's based on this evidence and arguments that this dose of 40 milligrams per day was validated and used by the DNDi in its anti-cough trial, because
Nicolas, you just need to speed up a little bit.
Okay. So this dose is currently used by is currently tested for fluoxetine in the DNDi anti-cough trial. Now, I have the pleasure to give the floor to Dr. Céline Cougoule.
Thank you, Nicolas. So here I would like to present you the preclinical data we obtained with fluoxetine. So just briefly, when Nicolas contacted me in March 2021, he had already in his hand all the observational data he just presented to you, and also some data already available in the literature for the effect of fluoxetine in vitro on SARS-CoV-2 infection. What was really missing at this stage was proof of efficacy of fluoxetine in animal models. So first, I would like just to quickly present you that fluoxetine in different cellular models inhibits SARS-CoV-2 infection. And this is also true not only for the original SARS-CoV-2 strains, but also fluoxetine presents effective activity against the different variants of SARS-CoV-2. And the effect of fluoxetine was also confirmed in a primary lung system based on human lung slice, where you can see here on 3 independent donors that fluoxetine is able to reduce viral load in this system. And this is also consistent with the dose which correspond to what's taken by the patient on a daily basis. So next slide. So first we started by confirming this in vitro data, and you can see that here at IPBS, also using virolytic cells, but also alveolar human epithelial cells expressing IC2, we confirm that fluoxetine is able to restore cell viability after SARS-CoV-2 infection and reduce significantly the viral load for all these 3 variants. So then we decided to move to the in vivo model. So first, just to show you also the data which are available in the literature regarding the EC50 for fluoxetine and fluvoxamine with a better efficacy of fluoxetine. So then we moved to the animal model, and we used humanized mice for expressing human ACE2 and epithelial cells. So it's a classical model for coronavirus infection, which was very useful also for SARS-CoV-2. So we had 2 groups of mice. Both were infected. One were treated with the vehicle and the other one with fluoxetine. But at this stage, it was very important to determine the right dose of fluoxetine to use in this mouse model. So next. So for that, we benefit the expertise of our colleagues in Germany, namely Eric Gulbins and Johannes Kornberg, who are experts in this plasma molecule. So they performed for us all the pharmacokinetic experiments to determine the half-life of the fluoxetine in mice. So here you can see that we have a very short half-life, around 6 hours. They also tested different doses of fluoxetine, and you can see that, as expected, there is an increase in the fluoxetine level in the serum of the mice according to the dose. And most importantly, they has the capacity to directly assess the efficacy of fluoxetine at inhibiting ASM in the lung. So this is what presented here. So you have acid sphingomyelinase activity in the lung of the fluoxetine treated mice. And you can see that with a dose of 40 milligrams per kilogram, we reach a significant inhibition of ASM. So we decided to perform our experiments with this dose of 40 milligrams per kilogram. Next. So the design of the experiment was very simple, I would say. So we had 2 groups of mice infected with SARS-CoV-2, and then we started to treat on a daily basis, so once a day, mice with fluoxetine by intraperitoneal injection of fluoxetine. And at day 2 and day 6, we sacrificed the mice to evaluate the viral load in the lung of the mice, but also inflammatory markers. Next. So you can see here that our primary readout was to measure the viral load in the lung. And you can see on this graph that at day 2 and day 6, we measured a significant effect of fluoxetine at reducing SARS-CoV-2 load in the lung. And also we confirmed that at the RNA level, at least at day 2. We were not really surprised to confirm that fluoxetine treatment trigger a body weight loss of the mice. This was already described in the literature. So we reproduced this, and it was after this strong body weight loss induced by fluoxetine, really complicated to really assess by measuring body weight the benefit of fluoxetine. But by measuring the body temperature, we observed at day 6 that the vehicle-treated mice were really in a kind of sepsis condition with a strong reduction of body temperature. And this body temperature reduction was less with fluoxetine, demonstrating an improvement of the clinical parameters of the mice under fluoxetine treatment. Next. So next, on the lung samples, we also evaluated inflammatory markers. Here we assessed the expression level by qPCR of IL-6, TNF-alpha, CXCL10, and CCL2. And these markers, as previously presented by Nicolas, are important inflammatory markers also observed in patients with severe COVID-19. You can see that under fluoxetine treatment, we have significant reduction of this inflammatory marker at day 2 for all of them, and also at day 6 for IL-6. So these data show us that in a mouse model of SARS-CoV-2 infection, fluoxetine has antiviral and anti-inflammatory effects. Next. Finally, we also took advantage of these experiments to evaluate the chiasma hypothesis in the mice model. And for that, the lung samples were analyzed in Germany by our colleagues, and they performed some sphingolipid lipidomics analysis, and they show that while the ceramide level is not really modified in the context of fluoxetine treatment, we observe a significant increase of exosyl ceramide, specifically the 16, and this leads to an increase in the ratio of this exosyl ceramide and ceramide. It's very important because in COVID-19 patients, this ratio is reduced with severe form of COVID-19. So here, in the mice model with fluoxetine, we can reverse these markers. Next. So I hope that I convince you that this chiasma effect has a potentially important role in the effect of fluoxetine to fight COVID-19 and to inhibit SARS-CoV-2 infection. And this pathway is a very complex pathway, very tightly regulated. So even if ceramides are central in all this pathway, it's still research for us to better understand more specifically which pathway are the most important in the fluoxetine effect. Next, and to finish, I would like to also highlight that not only SARS-CoV-2, I would say, use this ceramide pathway for infection. It has been described for other type of viruses and especially for the Ebola virus. Next. So I give the floor back to Nicolas to finish and conclude. Thank you.
Thank you very much, Céline. Just shortly, in terms of observational data, I try to summarize all published studies, and as you can see, all published observational studies are convergent to show a significant effect of fluoxetine. Effect, I mean, significant association with all-cause mortality, COVID infection, 28-day mortality, intubation, or death. So this association seems to be among the strongest among antidepressants. Now, about clinical trial results, we all heard of fluvoxamine in COVID-19. But first of all, why fluvoxamine? So the idea of fluvoxamine was from Dr. Angela Ryerson from Washington University. And she contacted Professor Eric Lenze for conducting a clinical trial. And very shortly after the publication of our preprint with the observational results, we've been contacted by this team and a broad team to discuss about the choice of the antidepressant to use in a first clinical trial. Clinical trial, as you know, that has been published in JAMA several months ago. The discussion was first that in terms of that fluvoxamine should be preferred because of the potential role of the sigma-1 receptor agonist effect. We know that fluvoxamine is better than fluoxetine according to a unique studies conducting in brain of rats, and that we have an animal model showing that this effect could underline the anti-inflammatory properties. At the same time, we had this in vitro data showing that chiasma effect could be interesting. In that case, fluoxetine is the best one and fluvoxamine the third one. The other evidence, other argument was that the blood half-life of fluvoxamine is lower, is shorter than that of fluoxetine, and that they had better hopes that it could reach a potential efficacy shortly after giving the first pill. But we know that the distribution, the target organ should be upper airways.
Nicolas, sorry, we need to. I would like to leave some time for the discussion. So sorry.
Okay, I will finish. Yeah, sure. So the lung distribution is important, and it's known for fluoxetine, which is higher than in the blood. We had observational data, tolerability showing that fluoxetine is well tolerated, and a question about location of production lines we had. So the clinical data of fluvoxamine, you heard about that, showed that globally we have several clinical trials that show lower risk of clinical deterioration and lower risk of hospitalization or emergency care of more than 6 hours. And especially the Together trial found a significant association between in a randomized clinical trial with reduced risk of hospitalization or emergency care of more than 6 hours that are still significant where considering more than 24 hours or the FDA criterion for severe COVID-19 as it has been done for other antiviral molecules. And what is interesting is the per protocol analysis. These trials of repurposed treatment are at-home trials, so the compliance is not 100%. And when examining just the population who correctly took the medication, the effect was stronger. And for example, for all-cause mortality in the Together trial, the reduction was 91% of death in the specific population that took correctly either the fluvoxamine or the placebo, and no significant differences in adverse events. So to conclude, we do have a substantial scientific presumption for fluoxetine, but we do not have currently clinical trial data and substantial clinical evidence for fluvoxamine. We do have concordant in vitro, including variants of concern, animal and observational data for fluoxetine, and several trials, including one large RCT together for fluvoxamine. The mechanisms of action likely are antiviral effects through the plasma effect with concordant in vitro, ex vivo, in healthy volunteers, animal, observational, and clinical data, and anti-inflammatory effect that still need to be fully explored to understand the specific mechanisms. The advantages, of course, it's the tolerability, which is well known, the immediate availability at low cost, that fluoxetine specifically is on the WHO list of essential medicines. It's easy to use, PEROS, and mechanisms that is likely not to be sensitive to variants. We know that it's an important thing. So at this stage, we think that there is an urgent need of large RCT of fluoxetine, and also to continue with fluvoxamine on dedicated platform for both out and in patients, and also as a potential prophylactic treatment of long COVID, given its well tolerability. I want to conclude and to also acknowledge the strong and the important work of all collaborators and all people we work with, and we wanted to thank specifically the DNDi staff and Nathalie Straub-Wergraft, who implement an arm in anti-COVID trial with fluoxetine. Thank you so much for listening until the end of our presentation. Thank you.
Thank you so much, Céline and Nicolas. Sorry. Very interesting presentation, which really illustrates exactly as you presented how you can go from clinical data to basic research and then go back to the clinic. So we have time for a few questions, and well, as always, we will look if somebody has questions on the. You can post them on the chat, or you can immediately show up. Maybe I will start. I mean, obviously, in the context of the global virus network, what you have shown on this pathway and these mechanisms is very important. And also, you have alluded, Céline, at the end of your talk on the fact that this might apply for other viruses. Do we have the same level of evidence for the other viruses? You see, I was curious to know whether, for example, you mentioned Ebola or others. Do we have the same level of evidence?
You mean the same level of evidence of the efficacy of the involvement of the viral replication? I think it's not at this stage, I think after the massive effort during the COVID pandemic, I think that we have more evidence for SARS-CoV-2 infection, but there is some evidence showing that there is involvement at different part of the pathway and different type of lipids that could play a role for other viruses. And there is some review and references on that, but I cannot quantify exactly the level of evidence. But yeah, I think all these ceramides pathway is essential for the regulation of cell membrane, clustering of receptor, and we know how host membrane are and homeostasis of cell membrane is important for virus replication. So yeah, it's central.
Thank you. You have a nice comment from Dr. Bonaguro, Franco Bonaguro. So there is a question, I don't know if you can see them. Are there any advantage of fluvoxamine or fluoxetine as compared to Paxlovid? You could not escape this question.
Yeah, I know, and this is what we are at the moment addressing, still in collaboration with the NDI. This is important information also they want to get, and we are still working and doing experiments to address this question. So we are comparing fluoxetine and fluvoxamine. We are comparing fluoxetine and the Paxlovid in the mice model, and we are also testing the combo of the two molecules. And we also have experiments in progress to compare fluoxetine and fluvoxamine. We don't have the results yet.
There is a question for Nicolas, I guess, from Franco Bonaguro. I wonder whether the fluoxetine dose is within the regular dosage used for psychiatric range.
Yes, absolutely. With the PK modeling, we found that an optimal dose could be 40 milligrams per day, and that's within the usual range used for depression, including in older adults.
Thank you.
I can just complement this question because our colleagues in Germany are also still working on this question, and the experiments they did with amitriptyline in healthy volunteers are in progress also with fluoxetine to confirm that the dose. Okay.
So I guess that we have to we are coming to the end of this conversation. But that was very good to have this presentation, and we hope that this will stimulate some support some of the ongoing clinical trials, and that we can establish a connection between you and some members of the GVN to go maybe further into some experiments regarding SARS-CoV-2. That was really the aim of this webinar. So, well, we have to conclude. So thank you so much, both of you. Thank you to all the attendees for these webinars, which are obviously an important feature of the GVN activities. Sorry, somebody is asking, can the slides be shared? See, which is a sign of interest, Céline and Nicolas. And the answer is depending on you, Céline and Nicolas. You will let us know.
Okay.
Okay. Thank you so much, and have a very good day, everybody. Thank you. Bye-bye.
Thank you for the invitation.
Bye-bye.
Thank you very much.
Bye.